Hello, and welcome to this video on the statistics and cause of autism spectrum disorders. Autism spectrum disorder is a collective term for a group of developmental and behavioral problems. Autism is characterized by impaired social interaction, impaired communication, restricted interests, and obsessive behaviors. The prevalence of autism is currently estimated at 67 children every 10,000, a rise from an estimated 5 in every 10,000 children in the 1980s. Autism covers three core diagnoses: autism as its own category, defined by poor function in all metrics used; Asperger's syndrome, defined by poor communication metrics but a high intelligence on the tests given; and pervasive developmental disorder not otherwise specified, a poor communication metric but not within the boundaries of Asperger's syndrome or autism. What follows is a short summary of the etiology and impact of autism. The other two diagnoses will be given independent videos, as the pathology of each is diverse and unique enough to warrant the detailed explanation of the development in each condition. The rise in diagnosis and greater publicity of autism spectrum disorders has given rise to three main challenges to modern science, medicine, and communities. The first is understanding how autism comes about and how it is caused. The second is dealing with the public conspiracy theories about its origins and causes, particularly the anti-vaccine communities who believe autism is caused by administering too many, the wrong type, or poorly formulated prophylactic medicines. Finally, how the medical, social services, and community can support and nurture those with autism. The latter two topics will be covered in sequential videos. To begin with, the scientific community has reached a consensus on several key points, and these are: autism is markedly hereditary. It has a prevalence of 5 to 20,000 cases per every 10,000 children, and the breadth of which can be explained via the changing diagnostic criteria, greater efficacy in identification, and reduced social stigma. It is caused by multiple genes which are distributed continuously throughout the population, and it is the interplay of these genes that causes the type, severity, and timing of autism. Autism is not caused by vaccines, and that there is no evidence for this. But briefly, a 2002 study by Madsen et al. Used data from 500,000 children. They found no increased risk of autism in normal and autistic children. Secondly, that there was no clumping of autism diagnoses after children were vaccinated. And third, no autistic spectrum was ever associated with receiving or not receiving the MMR vaccine. Next, that the interaction of genes and the microbiome further impacts on autism, and that autism may be treatable with a deeper level of understanding of the genes involved in the spectrum. Autism is a neurodevelopmental disorder with marked heritability. The neurodevelopment disorder is exhibited through behavior and cognitive symptoms rather than traditional neuropathological indicators. Autism has been shown to have genetic similarities with Fragile X syndrome, Rett syndrome, and more. Autism is characterized by poor social interaction, poor communication skills, repetitive behavior, and narrow areas of interest. This has led researchers to draw a comparison with other conditions, such as Angelman syndrome, where a similar lack of communication can be seen, the same degree of fascination with a single thing, and poor social skills. Autism is apparently mediated by alterations in the neural stem cell determination, timing, and pattern of neuronal growth and differentiation, synaptic function, and apoptosis, which lead to altered brain development. As a result of this and other observations, small-scale studies have been done. Over time, these gained traction as evidence mounted for a genetic cause. What was strongly indicated in these small-scale trials has now become a significant factor in future research. Through immensely large nationwide studies across several countries, including Denmark, Greenland, and Holland, large data has been gathered. What they have found is that the genetic likelihood of autism is higher in monozygotic twins at 60%. It's significantly high in siblings of autistic children, and it has a receding risk in relatives as they get further and further away in a family tree. This demonstrates the effect of autism phenotypes and the degree of severity as it relates to genetic dosing. As has been demonstrated, autism has a significant neurodevelopment and pathology aspect, much like Rett syndrome. Rett syndrome is characterized by repetitive motions and gastrointestinal disorders. These are common characteristics of autism. Rett syndrome is known to be caused by an inherited mutation in 5% of diagnoses, or a sporadic mutation in 95%. This is in the MECP2 gene that encodes for a protein enabling the proper function of nerve cells. Through this mutation, noradrenaline cannot properly innervate the brainstem, leading to central nervous system dysfunction. Keep this in mind whilst the autistic neuropathology is described here. Autism is characterized by the neuropathologies and the behavioral changes caused by this. The nature of the pathology causes changes in behavior, and this includes alterations to the signaling pathways, altered synaptic plasticity, neural migration, and brain anatomy. Under other circumstances, the neuropathology of autism could be compared with brain cancer, brain damage, or severe embryonic development problems. However, autistic individuals are widely varied, and with appropriate support can be functional, active members of society. There are several candidate genes that are being looked into: the GABRB3, UBE3A, MECP2, and others that will be described here. Two genes identified in 2003, NLGN3 and NLGN4, which make up part of the neuroligand family, are responsible for neural cell adhesion and neural synapse development. This led to a dysfunction of the excitatory or inhibitory function of these neurons. This particular mutation may account for up to 1% of autistic diagnoses. Another is fibroblast growth factor, or FGF, which has been identified in autism as hypothesized as being the underlying cause in large autistic brain volume. FGF has been proposed as being responsible for the greater number of cortical excitatory neurons and excess network excitability, leading to hyperreactivity and seizures. These are many of the phenotypes and risk factors associated with autism. The role and prevalence of these genes is given further credence by findings in the size and volume of the brain in autism. The clinical onset of autism is preceded by two stages. This is abnormal growth, with reduced head size at birth, followed by a sudden and excessive increase in head size between 1 and 14 months. Further research investigated the role of inherited genes with male mice. They had a dominant expression of paternal genes, and they demonstrated a larger body and smaller brain, whilst maternally dominant mice had the inverse. This type of mutation and consequent diagnoses is seen in Angelman syndrome, where the maternal UBE3A gene is the only actively expressed allele. This is called imprinting. When this is dysfunctional or silenced, the gene is not expressed at all. In Angelman syndrome, the children express a loss of language skills, intolerance to change, and stereotyped behaviors going into and after adolescence. In Angelman syndrome, UBE3A is the sole locus or cause of the diagnoses. In autism, ubiquitin is considered a significant risk factor, but not the sole cause. Interruption of the UBE3A gene is associated with disruption and poor regulation of the p53 gene. This has an effect on the Purkinje cells and is consistent with the pattern of brain abnormalities found in autism. The examination and acceptance of Angelman syndrome as a genetic and inheritable diagnosis has given considerable credence to the cause and mechanism for autism, particularly the role of imprinting and selectivity in gene expression. As demonstrated, autism has significant neural development and pathology aspects, much like Rett syndrome. Rett syndrome is characterized by repetitive motions and gastrointestinal disorders. These are common characteristics of the autism spectrum and diagnoses. Rett syndrome is known to be inherited in 5% of diagnoses, or due to a sporadic mutation in 95%. This occurs in the MECP2 gene that encodes for a protein enabling the proper development and function of nerve cells. Through this mutation, noradrenaline cannot properly innervate the brainstem, leading to central nervous system dysfunction. MECP loss leads to a significant change in the DLX5 gene, which has a crucial role in forebrain development. In the case of MECP, loss of its functional expression leads to an increase in DLX5. This causes a significant reduction in both initial and long-term neuron development and maturation. Mouse models of MECP with a doubled expression of the gene resulted in enhanced motor and contextual learning and neuroplasticity up to 20 weeks. Like in autism, there is a delayed onset of symptoms on which the diagnosis is predicated. As time continues, the condition is exacerbated. The similarity with autism is likely why it was included as an autism diagnosis until 2013. Conditions with a significant downstream mediator are not uncommon, as seen in this picture. Noradrenaline creates a complex series of outcomes from the variety of receptors it interacts with. The above genes have several common downstream targets. Particularly notable among these are the gamma-aminobutyric receptors, or GABA receptors. GABA is the main inhibitory molecule in the central nervous system. It also has a crucial role in the formation of the brain architecture. The GABRB3 receptor is the only GABA receptor present in all GABA complexes and in early life. This gives further credence to the possible role of GABA in the development and maintenance of autism. This has been reinforced by findings of abnormalities in plasma GABA levels, reduced GABA receptors in autistic brains, and pharmacological interventions targeted at the GABA receptors, alleviating autism to some extent. GABRV3 has shown a strong paternal bias in expression. Like with other disorders, the parent from whom the gene is inherited has an adverse effect in the right combination of other genes, further to which yet more of the GABA receptors and genes have been found with an imprinted effect and bias in autism. Serotonin is another strong candidate for investigation of autism. This is due to highly elevated serotonin in 30% of autistic diagnoses and first-degree relatives. This is where a population-wide distribution of genes becomes extremely relevant. Serotonin targeting drugs alleviating autistic behaviors, functional imaging studies of serotonin in autistic brains, the central role of serotonin in autistic behaviors, particularly repetitive behaviors, genetic association studies. These studies have found that there are several genes regulating serotonin transport, amygdala activation, and serotonin synthesis. These had further association with the degree of autistic symptoms. It is believed that this combination has an effect on early fetal brain development. These then have an effect on later brain development through the GABA pathways. There is an emerging field of evidence that the dietary and microbial population of autistic individuals can have an impact on the severity of autism. Particularly relevant are propionic acid and related short-chain fatty acids. Collectively, this offers support that gut-derived factors such as dietary or enteric bacteria-produced short-chain fatty acids may be plausible agents that can trigger autism or related behaviors and need further exploration in clinical science. This avenue of investigation shares many measures with the neuropathologies and relates to metabolites generated from diet directly contributing to the neurotransmitters of the central nervous system and the genes that express these. Although this video is by no means exhaustive, hopefully it has given some concept of how autism is inherited or means by which it may be caused. Some other topics that may be of interest to you are the male brain hypothesis, mirror neuron system issues, temporal lobe dysfunction, and mitochondrial dysfunction. Thank you for watching, and please post any questions you may have in the comments section below.